Hi everyone, Dr. Williams here. Today I'm going to go over avian diversity, which is covered in your textbook, the Handbook of Bird Biology, and Chapter 2. So diversity, of course, is really important. We have to understand avian diversity if we want to enact conservation. But there's a lot of bird species. There's been over 10,000 living bird species identified, and this is just one example of a phylogeny or a hypothesis as to the relatedness and diversification in birds. So you have hopefully studied diversity in other courses in biology so far, and you should be familiar with the term order, family, genus, species, as well as higher classifications. Classification is just that, how do we organize groups of species or individuals in relation to relatedness or morphology, that kind of thing. So classification is a process. It is still an ongoing process. So the phylogeny and the order, the taxonomic order of bird species has changed, and even the last 10 years. And there are still places where the phylogeny is unresolved, where we could possibly still be seeing movement between different areas. So as we get more evidence to help support the hypothesis of the phylogeny or the diversity, the classification system, then we will keep changing it. So it is a process that will be changing, and each year a classification scheme is put out, and sometimes they change orders or species, genera, that kind of thing, so based on new evidence. But this hypothesis then reflects our understanding of the relationships among these species. And of course, when we put it into a phylogeny, we're looking at it in terms of evolutionary relationships. So with bird names, birds have scientific and common names. The nice thing about birds is that there is a worldwide classification system, Eurocentric, so it does have English names. So the English names are all standardized by the American Ornithological Society Checklist Committee, and this is recognized worldwide. There are also local names, so it's important if you do go to, say, another country, there are going to be local names that you have to decipher. And thus we're going back and looking at the scientific names will be really helpful. We also have banding codes. So if you click on this in the PowerPoint, you'll be able to go to the U.S. Geological Survey, and they have a list of all the species in taxonomic order, and that also has the banding codes to go along with it. So make sure, if you're interested in anytime you're not sure what a banding code is, look it up. And again, we use these banding codes as shorthand. It's four letters, usually the first two names of the first word, and then, or the first two letters of the first word, and then the first two letters of the second word in the name, but not always, because there's a lot of overlap. So do use the banding codes, get in the habit of that, use banding codes in your field notebooks. So again, a phylogeny is a hypothesis of the relatedness or connectedness of different species, and there's a lot of different ways that we can think about how do we classify species. So there's a biological species concept that hopefully you learned in 1710, where it is based on a variety of factors, including not having offspring that are hybridized. So you can't interbreed. However, we do see interbreeding, and we'll get into that a little bit more, even with species. So biological species concept is really useful, but again, it's not all-encompassing, and we can get into that a little bit more. We can also think about phylogenetic species concept. So this is more looking at the history of relatedness and the evolutionary separation. So how long in time, evolutionary time, have these individuals been separated? But there are different concepts. So up here in the top right, I have these different species concepts through time, and these could be moved along anywhere in here. So phylogenetic species concept, this is based on DNA, so, and monophyles, so remember those terms from evolution. But this could happen anywhere in here. You know, at what point do we say that they are phylogenetically or genetically distinct enough to be a separate species? The biological species concept here, again, we have two species, species A and B. And here we say, well, they're species because they're reproductively isolated. Well, the thing is, though, a lot of species are or individuals are reproductively isolated because of maybe geographic barriers. But if you brought them together, would they hybridize? So that's a harder thing to answer. There's also this ecological species concept, and this is based on different habitats. And a lot of times, if you think about ecological species concepts, if species are in different habitats and they're breeding in separate habitats, they could genetically diverge because they are geographically or isolated. They could be isolated in time by breeding. Or they may not, just because they're in different habitats, they don't see each other, so they're not going to interbreed. So over time, they could become a new species just based on ecological separation. There's also this morphological species concept, thinking about how morphologically distinct and different individuals within are. And then a genotype cluster concept, when this is differences in allele frequencies. So not getting a whole lot into this, but basically, again, we're thinking about, you know, if you have this ancestral species, you get some sort of barrier to gene flow. So it could be ecological, it could be geographical, some sort of barrier. And you start to see selection and drift, and then the species may diverge. And at what point do you say they're different enough? But again, this is a process, so be thinking this as a process. It's really, again, difficult because some species can look so different from each other, but still be the same species. So this example was from your book, and here basically there's differences in coloration. These are both males of the same species, the Asian paradise flycatcher. Both forms can occur and interbreed at the same location. So they are the same species, but they look totally different from each other. In the birding world, we often joke because there are splitters and lumpers. And some people, this is really serious, because if you split species, then you get more species on your list. And there are people that go out and they just bird and they twitch. They want as many species on their list as possible. Maybe not as interested in the biology or the ecology of the species. But there are arguments on when do you split. When is a species different enough to actually be called a species versus a subspecies or a type or a race? So here I have two examples. This bird up top is the common yellowthroat. This is a bird that you could see in a lot of wet areas here, wet and shrubby areas. They'll make these sharp chips, so if you hear them, you'll learn their chip note, and you can find them pretty frequently. They're usually going to be low in the shrub layer. They do live, or they breed up here. This is in the orange is the breeding range. The purple is the migratory or all year, and then the blue overwintering. So they do breed in North America, in our area, and then they migrate. They can be found in Florida over the winter, and then the Bahamas as well as Central America. There's 13 subspecies of common yellowthroat and 4 distinct species, including one in Baja California, one in northeastern New Mexico, another one in central Mexico, and the Bahama yellowthroat that you see right here is a separate species. They could occur in the same area. If you look at their breeding or at their ranges, they could occur at the same area during the winter, but the breeding seasons are separated. So when our common yellowthroats are down in their wintering range, it's at a different time as when they're breeding. So you have separation in breeding through seasonality or time, so they are not interbreeding, and you've had separation, and there's enough genetic distinctiveness that they've decided to split the Bahama yellowthroat into a different species compared to the common yellowthroat. So again, the biological species concepts becomes really kind of tricky because we start to see hybridization. So we start to find exceptions to a rule. And here, we're going to start over here with the cerulean warbler and the northern parula. One of my he's a bander on my permit, Ryan Trimbath. He's a PhD student, should be finishing up soon up at University of Akron. And he called me one day really excited. He says, I think I have a hybrid. He had been telling me all season long that he's been hearing a cerulean warbler and a parula in the same area and some differences in the song. And then he actually caught one of these, the offspring that he thought was a hybrid, and asked if he could band it. Now, we're not allowed to band birds if we don't know what they are, if we can't identify them, but there are some exceptions for hybrids, especially if you can identify it that way. So I told him, sure, go ahead. And so he ended up capturing it. He took some feather samples and some blood samples and went up to the Cleveland Museum of Natural History, where he worked with Andy Jones to get the genetics to process the DNA. And he found that the cerulean warbler was the mom and the northern parula was the dad of this hybrid. So this is a hybridization of these two species that are usually in different habitats. We usually associate cerulean warblers with high and dry forests, oak forests in particular, whereas northern parulas are often more lowland. They're where there's sycamores, river systems, riparian habitat. So a lot of times they're not in the same habitat. They are of the same genus, though. They're both within Cetophaga. So we have hybridization within this genus. Now, even more strange is that one of the students with Henry Strebe, I believe, up at University of Toledo, they're studying golden wing warblers. And they found a hybrid that they thought was a hybrid between a cerulean warbler and a golden wing warbler, which you see right here, okay? Another hypothesis would be that it could have been the paternal was a blue wing warbler here. So we know that golden wing warblers and blue wing warblers do hybridize. They're both in the genus Vermivora. And right now, golden wing warblers are in serious decline. They're becoming more and more rare, and blue wing warblers tend to be taking over those habitats or are showing up where the golden wings are. Now again, cerulean warblers, forested habitat, oak forests a lot of times. Golden wings and blue wings are more in these shrubby, early successional habitats. But this bird they captured, they thought, it actually looks like a hybrid between those. And they did find, indeed, that the cerulean warbler was the maternal DNA, and then the suggestion was that the paternal DNA was a golden wing warbler. So here we have hybridization between species within two different genera. So again, the Cetophaga for cerulean warbler, and V here is for Vermivora for the golden wing, blue wing complex, so golden wing warbler, Vermivora. So it's not just within a genera that we see some hybridization among these individuals. So we're going to come back to this phylogeny. Again, a phylogeny is a hypothesis that is a representation of the history of evolutionary connectedness. And again, make sure you check out this, spend a little bit of time in the book. It's a little bit hard to see here, but in the book, spend a little bit of time looking at this and looking at the relationship and the diversification rates within each taxa. So some you see high diversification rates and some lower diversification rates. Also, make sure that you review terminology that you should have learned in evolution, including what nodes, branches, and sister taxa are. You should also be looking up terms like synapomorphy, plasiomorphy, that kind of thing. So just refamiliarize yourself with some of those terms from evolution. So here we can zero in on part of the phylogeny and look at Mimidae. So a family of that includes mockingbirds, thrashers, and catbirds. So up here we have a brown thrasher. This is a bird that you could see in Ohio. Same with the catbird. You'll be seeing those within the next few weeks, and then they're going to take off to overwinter much farther to our south. But we can look at where these are on the phylogeny to look at how related. So the gray catbird right here And gray catbird is more closely related to a white-breasted thrasher, which I've never seen. And then the brown thrasher over here. So the gray catbird and the brown thrasher, if we follow the phylogeny back, we know that they share a common ancestor. And we can also look at this black catbird then here. So we look at the morphology, how the black catbird looks compared to the gray catbird and brown thrasher, and we can follow this back on the phylogeny. So the black catbird, gray catbird, and brown thrasher all share a common ancestor. So this is a phylogeny based on 19 different genes that was published back in 2008, and this is again there's been studies that have modified this phylogeny since then, but this is, I think, really instructive because it is over all these genes. And what I want you to do, this is also in your book, I want you to take some time and explore the relationships among the different orders that I have listed here specifically. So find all of these orders on this phylogeny and look at the relationships. And I also want you to pay attention to these areas where we have some gray areas. So they have these dashed areas in here, and you're going to see later on when I have apodoformes with our hummingbirds and swifts, some places keep them as apodoformes, but others are keeping them within capromongiformes. And I think more current hypothesis is that they are within the capromongiformes, not their own order. But anyway, so explore these relationships, spend a little bit of time on this. These are common orders that you'll be expected to know, and try to understand where they fall out and relatedness to each other. So your book actually does a really good job talking about this, and I just wanted to highlight it because, one, I really, I think how unfortunate we are that we have not gotten to see some of these birds that have gone extinct. The moas are an example of some species that went extinct a long time ago, and we find these fossil records from New Zealand. There were between 9 and 11 species that they looked at. What was really cool, they were able to use the DNA to actually identify that there was sexual size dimorphism. So they thought there were more species there just based on the bones, but when they actually got into the ancient DNA, they realized, oh, males were actually smaller than the females in a lot of cases. And some of the information that they can get from DNA suggests that maybe males might have been incubating because they found the male DNA on the outside of the eggshell. So again, I'm not going to go into more detail on this, but again, make sure that you look through and read the boxes in this chapter because there's a lot of cool information in them. So you could imagine as an explorer in the 15, 16, 1700s, even the 18 and 1900s, you're finding birds and you don't have genetic information, you don't have DNA. So you're classifying and categorizing species based on other species that you already know and how similar they look. So here we have two birds that your book talks about in figure 211 that they look very similar. They have some of the same types of fly-catching behaviors, but they are not genetically related. Morphology can also be misleading. So morphology, we know, can change very rapidly. There were examples with the Darwin's ground finches, medium ground finches with drought. The bill shape changed very quickly due to natural selection when there were droughts, when only hard big seeds were available. Bill size increased very rapidly because any bird without the larger beak died. So here is an example where, again, morphology can be misleading. So this bird was named Hume's ground jay. Kind of looks like a jay. If you think about a blue jay that you might see here, they thought that maybe it looked like other jays in the family Mimidae. But when they got genetic evidence, then they found out that no, this was indeed more related to the tits and chickadees and titmice, so the family Paridae. So now it's been renamed as the ground tit. We can also be thinking about geography and how geography affects gene flow. So this example up top here with the Arctic warbler, they are distributed across northern Russia. They are pale Arctic distribution. And the blue dots are all genetically related individuals, okay? But we start to see some differences and divergence in the populations when we come down into these islands. So the gene flow is not happening with these populations, so they're becoming more genetically distinct. We can also think about how dispersal affects divergence. And here, if I move this out of the way, hopefully, we can see in this lower graph, this is your figure 215 in your chapter. They looked at 40 different South American species and looked at barriers, including the Andes Mountains and then also rivers. So these rivers are very large, and a lot of individuals can't or don't want to cross that large of an expanse because they could be susceptible to predation when they're out in the open like that. And they looked at it in terms of divergence and in terms of their dispersal ability and where they forage. So canopy species, if you're flying around the canopy, you might be flying more, you might be more used to these open areas that you have to fly through, compared to individuals that are lurking along in the deep understory where they're more hidden and it's shaded. And they did find indeed that genetic divergence was greater in understory species, so they're not able to disperse as much across the Andes. So understory birds aren't dispersing as much across the Andes. Genetic divergence was lower because of gene flow and canopy species. We also saw this across the Amazon River and across the Madeira River. So when we think about global patterns in avian diversity, there's a lot of factors that we need to be thinking about. We can look at ecological factors such as resources and how heterogeneous habitats are. And sometimes you're going to read things in terms of niche availability. My personal and a lot of people's opinion is that niche should only be referred to as a property of the organism, not of the habitat or availability. So be very careful with how people are using niche so that you understand what they're using it for. But if we think about resources available, the more resources that are available and the more heterogeneous habitats you have, the more opportunities you might have for individuals to be in there, and you might have more species there. Also, how much competition is there between different individuals that could force divergence so that maybe behaviorally or morphologically you get a little bit of specialization so that you're not competing as much. When we think about biogeography, I'm coming back here to the theory of island biogeography. Just to review this, hopefully you have all seen this. This is looking at species richness and rate of immigration or extinction, and looking at the number of species that are available on islands that are small and far. So you have fewer species, you have more species on small but large islands, followed by small near islands. If you're closer to a continent, you might have more immigration and rescue of small populations. And then the largest islands that are close are going to have higher species richness compared to farther islands. So just a review of the theory of island biogeography and how it relates to species richness. But basically, we also can think about how diversity increases with area. If you have more space, you have more room for more individuals. If you're thinking about also a larger area is a better target. So if you're a bird and you're flying along and you're looking for a large piece of habitat on the continent, then you might fly down to that versus a small patch. Same thing with islands. If you're flying along and you have a choice between a large island and a small island, you're probably going to choose that larger island because in general you know that there could be more resources on the larger island. Also, when you have larger populations, you have lower extinction probabilities. So again, this is more your population dynamics and population genetics that you need to be thinking about. So how do large populations, what are their probabilities of survival compared to low or smaller populations? We also have a generalization where diversity is greater near the equator than towards the poles. So this is referred to as the latitudinal diversity gradient. And there's a lot of reasons in this and a lot of hypotheses of why this is. It's a generalization, so there will be exceptions to this depending on where you're at. But again, in general, there seems to be greater diversity near the equator than near the poles. Okay, then I also have islands have fewer breeding species than continental areas of the same size, and that goes back up into this larger populations, and then also the rescue effect, where if you have mainland populations and you have dispersal, you could have individuals rescuing that population, even if it is possibly a sink area. Okay, so this, I wanted to go into a little bit more detail on this latitudinal diversity gradient. And this is from a paper by Saup et al. That was published in September 2019. They did some simulations to see what would be expected, and they looked at different, and again, if you click on it, you can go to the paper itself. The link is on the figure. But they looked at it with birds, mammals, and amphibians, and here we have this heat map of number of species, with the hotter reds being greater number of species. And in general, across all of these taxa, you start to see this difference, and I think I just went to the paper. Okay, I did click on it, and it does take you to the paper if you want to read more about it. But in general, I think we can get the gist here. So this basically is number of species along this x-axis in latitude here, with 0 being the equator across here. And with birds, we see increase in species diversity near the equator, and we get decreasing diversity as we go to the poles. Same patterns with mammals and amphibians in general. So why? Why could this be? So basically the tropics do have higher ratio of speciation to extinction than at higher latitudes. So that means that the number of species tends to increase over time in the tropics more than in at the poles. We also have a lot of ecological diversity and heterogeneity in habitats in the tropics, and that is a lot of times due to energy inputs, more solar radiation, and then also precipitation. And again, greater turnover in the tropics, which has to do with the speciation to extinction rate. So, we can think about biogeography and the realms, and we are going to be using the terms for these 8 primary realms. So we have near Arctic, Palearctic, Neotropical, Afrotropical, Indo-Malayan, Oceanian, that includes New Zealand, and then Australian, okay? So I want you to pay attention when I put orders up and look at the distribution. I often include the range map, so I want you to understand whether how different orders are distributed. Are they worldwide, or are they limited to some of these areas? And you're going to use these terms when you're describing their distribution. But basically, these biogeographical realms have a distinct avifauna. And a lot of times, these avifauna have become distinct because they are separated by barriers to dispersal. Down here, they talk about Wallace's line. So there's a really deep oceanic valley, basically, that has been a barrier to dispersal. So even though these are pretty close together, they're separated by this deep ocean valley and the currents that tend to keep these populations of avian diversity distinct. We also see that with, say, mountain ranges, east to west. So we have in North America, our mountain ranges tend to be north-south. So if you think about the Appalachian Mountains and the Rocky Mountains, but in, say, Europe, a lot of these mountain ranges are more across the latitudinal. So it would be a barrier dispersal from going north to south. So be thinking about what kind of geographical barriers that could keep these populations distinct. So that gets us to thinking more about these avian distributions and how to use these range maps. So you do have to be careful when you're drawing a range map, especially when you're doing large areas like the entire Earth. we can't be really specific in pinpointing where these birds specifically are. So we're painting a broader picture of where they might occur. So this is where you have this where they might occur in geographical space, and then you have to have knowledge of the species habitats so that you can go out and find them. So these species or these birds will only, for the most part, be found in the habitats that they require. So a few species that are very widely distributed include the peregrine falcon here, so worldwide distribution. It has the broadest breeding distribution of all land birds. So again, on these range maps from these are all pictures and maps from Birds of the World through the Cornell site. But basically, again, you've got breeding. So these are breeding in the near Arctic and pale Arctic. And then they overwinter in the neotropical, Afrotropical, Malaysian, and Australian areas, for example. The barn owl is also very widely distributed, and here we can again see this range map. This is based on eBird data, where these occurrences of barn owls are. And the nice thing about the eBird data is these are actually observations. Now you do have to be careful with eBird data because, of course, there's going to be more birders near more urban centers or near areas that people like to go more. So anytime you're looking at eBird observations, you have to be a little bit more careful about thinking about, well, who's actually going out and birding in those areas. Then we have the barn swallow, and this is a Passeriforme in the family Hirundinidinae. And here again we see a near Arctic and pale Arctic breeding distribution and then overwintering in the southern hemisphere. Again, we have to think about how, yes, we have these range limits and we draw it on the map, but we have to think about more than just habitat and availability. We also have to think about the physiological needs. And this is a really cool study published, I think, in 2019, maybe a little bit earlier, by Tingley et al. And Stephen Beisinger, who's an amazing ornithologist. They basically were looking at historical records. Over 100 years of monitoring in the Sierra Nevada, we had these different study sites with transects throughout these. And what they really did find is that there were shifts in breeding areas based on temperature and precipitation. So in this figure B, basically this is what this figure 1 is explaining if you want more detail. But when you looked at historical records, that would be the beginning of these arrows. That would be the historical habitats with the type of precipitation and temperature that was in those habitats. And then out to where the arrows are pointing, it's where that precipitation and temperature is now, where they're breeding now and what that historical habitat was like. Now, that's all good. That's showing that there's shifts, but I think it's a little bit easier to be thinking about it here. So here we have temperature and precipitation still. We have these black areas. So this is the geographic range that the bird could be found in, okay, at time point 1 and time point 2. But then we have the climatic requirements. So if the birds need what's in temperature and precipitation within these ranges, we would see that in this geographical region, the populations at site 2 and 3 would be living, but because 1 and 4 are outside of the temperature and precipitation areas that the birds need, they would not be there. But when we see changes in climate over time, we still have this geographical area, all right, but we see that there's differences. Well, there was an absence of species here before, and there's also an absence of species at the same site after that temperature precipitation has shifted because of climate change. This other part here, we can look at population 2. We see that, yes, it was in its thermal and precipitation ranges here, but when climate shifted, that precipitation temperature was no longer suitable for that species, so that population went extinct. We can look at population 3. It stays the same. It's still within its thermal and precipitation tolerance levels. And then the site 4 down here, we see now is within the geographical range, and it is now a habitat that is suitable for the species. So where site 4 didn't used to have birds, now we actually have birds using that space. So again, be thinking about what niche is, and niche should be defined as the property of an individual, not just empty habitat. You can think about what habitat is available, what resources are available, so food, shelter, thermal and precipitation tolerances. And again, there's a lot of different physiological tolerances that you need to think about. So thermal and moisture, precipitation I've talked about here, but it could also be salt tolerance. What about wind patterns, that kind of thing. Especially with some species like the black vulture, their distribution is moving north. They rely on thermals. We'll talk about this more later, but they rely on thermals so that they can get these uplifts and help to fly and stay aloft. But as climate is warming, they have been able to shift their distribution northward. This has way too much on it, but I did want to give you an idea when we think about the diversity of birds, just how diverse birds are. Again, 10,000 species. If we look at the order Passeriformes, Passeriformes has approximately one half of all living species today. And again, don't get too caught up on this. This is just showing the paper. If you want to look at it more, there is a hyperlink in the PDF or the PowerPoint that you can go look at. But it's just a stunning array of diversity. So a lot of birds are passerines, or we call them, they're perching birds. So a lot of them have song that's more complex, but they are perching birds. And this is what most of the birds that you see outside are, and most of the birds that I capture for banding are within the order Passeriformes. But there's a huge range of diversity in birds. So Opisthecomiformes, the Watson, is the only member of that order. So it's really wild to think about. You have all of these species within an order on this last slide, but then we have in this order, spell it correctly, don't worry about saying it correctly, but this will be an order that you're required to know. And also its distribution. So Watson is distributed in the Neotropical bioregion. We can also think about diversification rate. So when you have an empty habitat and you have individuals that move in and colonize that habitat, how fast do we see diversification happening? It tends to be faster after island colonization. There's lots of examples, including the Darwin's finches. Here I have a figure of the Hawaiian honey creepers. These are, again, orders Passeriformes. But if you look at them, it would be really hard to say what family they're in based on some of their characteristics. If we look at this Laysan finch here at the top, though, we can see, well, that looks finch-like, okay? But some of these other ones, not so much. So that might be like looking warbler-like, and who knows. But we do know, based on genetic analyses, that these Hawaiian honey creepers are in the family Fringillidae. Okay, so we have to think about what their common ancestor was, not just basing classifications based on morphology alone. So why would we see faster diversification rates on islands compared to elsewhere? Well, basically, we often tend to see this early pulse and then these slower rates over time. This could be because, again, you're dispersing into new isolated areas. There's a lot of resources to be exploited. So you see, as individuals start being able to take advantage of all these resources, that you see this rapid diversification. And it can also be because as you increase your density, your competition is going to increase. So if you're in symmetry, you would have competition among other birds that are with you, and it would be good if you could exploit different habitats or resources. And there's this thought that density, diversification rates could be dependent on density. So again, the higher density might have lower diversification rates because maybe we already saw historically that we had this rapid diversification. Here we can think about diversification in one order, the Charadriiformes. These are a lot of our shorebirds that we might look at. They include jacanas, snowy sheathbill, the Eurasian woodcock. So we have an American woodcock that you might still find here. Again, they will be migrating this fall, but they can be found in wooded habitats. We have the puffin. This is specifically the Atlantic puffin. We have a blacksmith lapwing. There's other lapwings. We have gulls that you might have seen. The ring-billed gull is one of those, and that's in the family Laridae. And this again is where hypotheses changed based on new evidence, right? So this black skimmer used to be in this Rhynchochopidae, but now it's in the family Laridae, okay, much more closely related to gulls. So how did we get birds? What is the evolutionary origin of birds? So now we're stepping back farther in time and looking at this phylogeny from way back in the Paleozoic. So we have these base reptiles. If we look at the extant groups, we have mammals, turtles, snakes, lizards, tuataras, birds, and crocodiles. So basically the reptiles over here, and then mammals over here. So if we think about going back farther in our classification, birds are in the kingdom Animalia. They are in the phylum Chordata. The class is Reptilia, okay? And the characteristics that birds have in common with other reptiles include that they have a single occipital condyle, that bone that your head's moving around on. So it allows your head to turn a certain way. We have a double occipital condyle in mammals. Birds also have nucleated red blood cells, okay? They have a lower jaw structure that is similar. They have a cleidoic egg, so this egg that's encapsulated that prevents desiccation. The female is heterogametic. So in mammals, male is XY genetically, whereas females are XX. In birds and other reptiles, the female is the heterogametic sex, so they have a ZW chromosome. The ankle, the tarsal bone structures are very similar, and then also lungs are very similar. We can actually trace back lung morphology with birds and crocodiles back to an earlier ancestor. So birds are within, if you go a little bit more deeply, in the group Archosauria. Okay, they're archosaurs. And archosaurs include this birds and also crocodiles. So if we go back on this phylogeny, we can think about sister taxa. So the only extant sister taxa to birds, or the most closely related sister taxa, are the crocodiles and alligators, Crocodilia. So both birds and crocodiles are archosaurs. And even more recently in time, birds, our hypotheses include that birds are descended from theropod dinosaurs, and more specifically the Maniraptora. So these little small, fast chasing, probably chasing insects, predatory type birds or dinosaurs. Same thing sometimes. So let's get into this theropod dinosaur. So the theropod dinosaurs and our modern birds had a lot of shared traits. So we do know now from fossil evidence that there were feathers on many dinosaurs, or the theropod dinosaurs. There's some suggestion that some of these feathers could have been used for flight. And how we know that, basically, we look here at a corncrake, we look at Archaeopteryx and the Weka. This is a flightless bird, so its primary feathers, or its wing feathers, are symmetrical. So this bird we would not expect to be able to fly based on its wing shape. The corncrake can fly. It has this asymmetrical wing, or feather shape, and this bird can fly. So a lot of the passerines that we study too also have these asymmetrical feathers. And Archaeopteryx had an asymmetrical feather, so that suggests again that it could have been modified or used for flight. We see evidence of pneumatized bones, so there's air spaces in the bones, and Dr. O'Connor, that has an office over in the Life Science Building, he had done a lot of study with looking at pneumatized bones and the lung structure of basal dinosaurs, Archosaurus, and modern birds. And so this is just a figure from one of his papers showing the pneumatic invasion of this pulmonary system. So basically these air sacs going into these bones in a great skua here, and looking at it at a different cross-section. Another paper that Dr. O'Connor published in 2005 indicated that Majungotholus also had air sacs. So these colored areas in here are suspected to be air sacs. There's evidence that they were air sacs, and much like the lung system that we see in modern birds today. So when we get into the respiratory system of birds, this will make more sense. But in general, the take-home point today is that these theropod dinosaurs had many traits that were similar, including these air sacs and the respiratory structure of the air sac system. Birds also have a small genome relative to other extant taxa, and we see that also in theropod dinosaurs. Their physiology and active lifestyle suggests that theropod dinosaurs were endothermic. They were bipedal, and you can see that here. So this is Archaeopteryx here, and a pigeon or a rock dove skeleton here. So we have bipedality. There's evidence from the fossil record that there was very rapid growth in the young of theropod dinosaurs, and that's again what we see with birds. The calcified structure of the egg was also similar in theropod dinosaurs to birds, and then behavior. There's evidence that the foraging behaviors and then even incubation and parental care behaviors are very similar in some species or some theropods with birds today. So we can't teach ornithology without talking about Archaeopteryx. Now, Archaeopteryx is not a direct descendant of birds. It's an offshoot that died out or became extinct. But it was one of the first fossil evidence that dinosaurs had feathers. So Archaeopteryx, this is one of the fossils that was fixed up from it. And you can definitely see the outlines of feathers on this bird-like dinosaur. So the bird-like traits of Archaeopteryx includes the plumage structure. Again, when I talk plumage structure, one of those examples is those asymmetrical primary feathers out here on the wing. They had fused clavicles. So a fused clavicle up in here, like the furcula. They did not have an expanded keel like our modern birds do, but you can see that the keel here in black versus the keel here on the pigeon. But they did have fused clavicles, this furcula. And then also some structure of the hand. We see this reduction in the bones of the hand, or that becomes the wing, right? So the wing is off of these hand bones and the forearm bones. But later our modern birds have these fused bones of the hand. Non-avian theropod traits include that Archaeopteryx had teeth. Birds have lost teeth because, again, with flying, teeth are very heavy. So they lost their teeth. Archaeopteryx also had a long tail, and they had claws. When most modern birds don't have claws on their wings or hands. So now we can kind of root ourselves in time and think about the Mesozoic birds and the Cretaceous. On the last slide, I had a little bit of that timeline too. And we can think about this phylogeny here. So here we have the class, we have Aves within Reptilia. Okay, so here we have on our phylogeny Archaeopteryx here. So there was a common ancestor of Archaeopteryx and our modern birds, but modern birds are not descended from Archaeopteryx. We see where we get a pygostyle. So this is this short tailbone that we can see on, you can look at it on the pigeon on the last slide. And then the Ornithothoraces here. And we see this diversion here. This is where I'm looking at now. So we had a group called the Enantiornithes, and these were also referred to as the opposite birds because some of their morphological structures were kind of opposite of what we see in modern birds. So they're not direct descendants of birds, but they were an offshoot of birds that were extremely diverse in the Cretaceous. And this picture here just shows some of that diversity. So they were flying birds. They had feathers. They had an allula, so their thumb feather, or not really a thumb feather, but basically the allula is this outer feather that they're able to pull out at the end of their wing, and it changes how the air flows over the wing. It disrupts, or it's used whenever there's turbulence so that they don't stall out like an airplane might stall. So the allula allows the birds to stay in flight at different speeds. And so the Enantiornithes had an allula, which is really cool to think about. Then we have, and that's again a trait that we see in modern birds as well. So their common ancestor may have had an allula. That would be a hypothesis we could make. Then we can think about these Neornithes, this rest of these birds. So here we can think about Confuciornis that we see up here in figure 229. There's a reconstruction based on fossil evidence of what this bird might have looked like. We see that it lacked a keel, not here, but it did not have that keel, that enlarged sternum for flight muscles to be on. It did have asymmetrical flight feathers, so out here we can see these feathers are asymmetrical, and again, that's an adaptation for flight. They had a pygostyle instead of a long tail, so that tail was reduced to this pygostyle that these tail feathers would have been on. And what was really cool, if you think about it, if you look at this bird, you might think, well, what are these long tail feathers for? They're probably not functional, but this is suggestion that there could have been sexual selection acting on this trait. So here we have evidence possibly of a sexually selected trait way back in the Cretaceous with Confuciornis. A little bit easier to say is Hesperornis. This is a fossil bird that was found in western North America, as well as Ichthyornis up here. But Hesperornis, these were swimmers. You can see teeth for sure. Their wings were reduced. They were much more body style like a penguin might be. And they had these lobe feet. So again, adapted for swimming. So they were predatory, probably fish eaters in the seas. And then Ichthyornis, if I was looking at this, I would be like, oh, that looks like a tern related to gulls. And again, these fossils were found in western North America. Again, suggesting that they were strong flyers, fish eaters, and again, their wing structure and sternum were similar to modern birds. And again, these were alive in the Cretaceous. So we kind of went through this area down here. We had the Enantiornithes. We had Archaeopteryx, some offshoot in here. These have all died out. We look at the Paleocene here, 65 million years ago, and this is where we start to see some of the splitting between the Paleognathes and the Neognathes. Okay, so two groups of modern birds and aves. When we think about this again, the origin of modern birds, and we put this again on the timeline, so we have the Cretaceous, and here we have the Hesperornis, and here the Ichthyornis up in here. So we see that these lines died out. We saw extinction of the dinosaurs, except for birds at the KT extinction, that big event, probably the asteroid hitting the Earth. But again, that's still a hypothesis. So anyways, we can look at this and see what birds have lived. So here, if it continues out, we see that these are the extant taxa, while these have all gone extinct. There's only one known fossil from the Cretaceous that is related to living birds. So we have a lot of holes in the fossil record that actually link to our living birds. So this is really worth kind of thinking about. This Vegavis fossil was found, and it was in the Anseriformes. What's really important here is that here Vegavis died out, but it had a common ancestor with the Anseriformes, these ducks, geese, and swans, here in the late Cretaceous. We also know from genetic information and dating that the Anseriformes split later than at the Gallo-Anseres. So the Galliformes split back here, so we can start to reconstruct the timeline of when divergence occurred. And then that split was more recent than with the Neognathes and the Neornithes split back here. So where we had the Paleognathes and the Neognathes in this group over here. So we can think about, yeah, the ostriches, emus, and kiwis split from the other modern birds way back in the late Cretaceous. So let's look at the Palaeognathes. And again, these are mostly flightless birds. So this group of modern birds, and we are going to need to be thinking about what these orders and the relationships are of these birds. So with the Palaeognathes, we have the ratites and tinamous, mostly southern hemispheric birds, and the extinct taxa in that includes the moas and the elephant birds. But the orders and families of these taxa for the Palaeognaths include the ostriches, the Strathioniformes. Strathionidae is our ostrich. We actually have two species in this, two species of ostrich, and their distribution is afrotropical. We can look at the rheas in the Rheaformes, Rheidae. There are two species of rhea. I'm not going to test you on how many species, but I do want you to know the order, family, and the distribution. So if we look at the range map, I pulled out South America here. So these are neotropical distribution for the rheas. The ctenoforms, I'm skipping around down here. So the ctenoforms, the tinamous, this is the only group that has members that can fly in the whole Palaeognathes. And again, they're neotropical in origin or neotropical distribution. The cassowaries here, okay, these are the cassowaries and emus. And here we can see that they have the Australasian distribution. And then, really cute, I want to see these someday, the kiwis. These are in New Zealand, okay? So those would be the Oceanian distribution, not just the Australasian. Okay, the rest of the extant avian orders are what we're going to be focusing on for the most part throughout the semester, and these are a lot of the birds that you're going to be seeing. So Neognaths, I'm going to leave it here that they are all living birds except for the Palaeognaths. So if it's not one of those orders on the last slide, then you're in the Neognaths, okay? Now, what I did on the remaining slides is I went through using birds of the world and pulled out the distribution for the orders and families, so mainly just for the families, I should say. So for weeks 1 and 2, if you would use the drop-down list in your Excel sheet that we gave you of the birds that you're supposed to find, the following slides have representatives of those groups. So here we have the Anseriformes. The family is Anatidae. There are 53 genera, so if I would have kept scrolling along this page, I would have seen more of these genera, but basically these pictures are just the thumbnails of some of the genera that are represented. Here we have order Columbiformes, Columbidae. These are our pigeons and doves, and again, you can see the distribution. We can keep going through here. This is where we have the Caprimulgiformes. Okay, up here we see order Caprimulgiformes, Caprimulgidae. These are our nightjars and allies, and the Nearctic and Palearctic breeding distribution. Here we have Trochilidae, the hummingbirds, and the Potidae, the swifts. And with the birds of the world, they have updated as 2020. Their order is Caprimulgiformes, although you'll often see it referred to as Apodiformes. So I might ask you that. So more recent taxonomy, there's some questions on where they belong. Are they Apodiformes, which means little or no feet. Feet are lacking. Both of these groups have very, very tiny feet. But they are actually currently lumped in with the Caprimulgiformes. And you can kind of go through all of these. So here with Cathartidae, the New World vultures, there's only 5 genera, so this is why you have 5 thumbnails of these. So if you have an arrow going forward on, say, like the Ardeidae, you see that there's a lot more genera that aren't pictured here. So please kind of go through there. And more importantly, as you're looking at these, so these are families and orders that you are required to know. If you go to your textbook, let's see if I can make this big. If you go to your textbook, okay, at the end of chapter 2, all right, there is this box, 2.09, and it's all these little kind of orangey pages. They have the orders and families of the birds of the world, and I did not copy this because you have it in your book. So you will be required, in addition to knowing the orders and families and being able to spell them correctly, you will have to learn the family and order characteristics that go along with it that are in your textbook. Okay, so make sure you refer to your textbook, chapter 2 at the end of it, throughout the semester. Okay, so anytime you get a new order or family, you're responsible for the information in your textbook on the characteristics of those orders and families, as well as the distribution of the order and family. Okay, so I'm going to leave you at that, and happy birding. I hope you go out and see lots of cool things. Thanks. Bye.